either in the chat or just press your space bar and tell me you can see me and hear me. Thank you very much. Can you see the screen? All right, good. Okay. For those of you that haven't had me before, I'm Dr. Phillips, our Professor Phillips, not Mr. Phillips. I'm a retired surgeon. I was a plastic surgeon some 20-something years ago, and I've been retired. I've been teaching at FSCJ for over 5 years now. After I retired, I played a lot of golf and then got tired of just playing golf, and so did something else. Taught a couple of years in high school, the hardest thing they ever did in my life, for the least amount of money. And then came to FSCJ and been teaching anatomy and physiology 1 and 2. And then occasionally, if they need me, I'll teach Chem 1020, Chem 1025C, and Chem 1032C. It used to be in the summer when we were doing face-to-face meetings. This has been the spring, summer, fall, spring, so forth. Spring, summer, fall. This is fall, spring. A number of semesters online. Obviously, this course is migrated towards being taught online. This is a face-to-face class. You're expected to be in the class online, listening to the lecture, live lecture. For the first few lectures, I'll record. There is a recorded lecture, except for Chapter 17. Chapter 17, I had to borrow the YouTube lecture, but for any lectures from 18 on, when I did 17 a couple of semesters ago, I saved it on Zoom, and I haven't been able to get it off of Zoom yet. But as a general, I'll record the first few lectures. But there is a recorded lecture. Some of the times it's from the last semester. Some of the times it is from this semester, like Chapter 18 here. This is the one I did last week for the other class. I have 2 classes now, I have 2 second semester classes. One meets Monday and one meets Thursday. And so we'll talk a little bit more about that. But this is what you come to when you log into the thing. And this is our work center here. Before you ask me where something is, look at the start of course documents. Before you ask me where something is, look at the start of course documents. Because the 3 big things you're interested in at the start of any course are the calendar of activities, the syllabus, and student registration for Pearson. And guess what? They're in the start of the course documents. If I put anything else in here, I will tell you. There's some also redundant stuff, lab safety requirements. We don't have to sign that anymore. Here's also your student contract. We'll talk a little bit about that, but that's an extra credit assignment. You have 2 weeks to download it, sign it, load it back up, and put it underneath assignments. And it tells you here how to upload assignments. I've even got a written instruction for video instructions for written instructions for uploading assignments. Okay, but this is where you start to look for anything. Okay, and I know the student registration works because I've already had 3 students sign up for it. Okay. And I get the student registration right off of the course that I create, so I know that it comes from that course. So if you can't sign up and it tells you it's the wrong number, okay, it's not the wrong number. It's something either you're doing wrong or Pearson's doing wrong. So you need to contact customer service. If you have a problem, just let me know. But you should be able to register on Pearson. If you're waiting for funds, you have 2 weeks to register. I do not look at the chat, okay? I do not look at the chat for questions. So you got a question, press your space bar and ask me a question, but I don't look at the chat. I'll look at it at the end, but I don't look at it going forwards. Calendar of activities is your roadmap, and is your roadmap for what's going on here. And this is your calendar of activities. It tells you what we're going to do this week, what chapter we're covering, what the Pearson assignments are, what the lab assignments are, when the Pearson assignments are due, and when the lab assignments are due. And so we have, because this is a 12-week class and we have 13 chapters to cover, we have 1 week where we cover 2 chapters, okay? And that will be when we cover lymphatics and the immune system, which is actually an ideal time to cover 2 chapters. Otherwise, we cover 1 chapter at a time. Most of the modules have labs. This is subject to change. You know, we could have hurricanes, tornadoes. Shouldn't have hurricane. Something could happen. So it's always subject to change. We could get behind, but we don't have very much wiggle room to get behind. So you have the notes, and you have a number of different aspects to move this course forward, in addition to lectures I've either already given or I'm given at the time. If people quit showing up for the lectures, number one, you're required to be here at the lectures. If you quit showing up for the lectures, I'll quit recording these lectures, and what you'll miss out on is the announcements. Okay, primarily that's why I record them. But this is the calendar of activities. It's use registrations right here. You've all done Pearson before, so you know what it looks like. I'll just click up here because I have trouble reading it. It's the usual thing, what to do. If you have a code you used last semester, it should still be good. And so that's what's going on with that. So we look at the student ratio. This is a big thing. I am very, very, very big on announcements. Let's go back. Go back to our modules here. Number one, Pearson is not hooked into Canvas. Pearson is not hooked into Canvas. And so if you get if you're one of those people that says, hey, this assignment wasn't posted on my Canvas shell, I'm sorry, okay? I am telling you, Pearson is not hooked into Canvas. You have 2 things to tell you what your assignments are. Number one, you have your calendar of activities. Number 2, you have in announcements, you'll always get 2 announcements from me. Usually you'll get this probably on Sunday or Monday. My other class is on Monday, so I work on their stuff on Sunday. So I'll probably work on your stuff on Sunday too. You'll get your login instructions to tell you how to log in. Obviously, you know how to do that, okay? You'll get your login instructions to tell you how to log in. And the other thing you will get is this week 1 announcement. It doesn't look like much now because we're just starting, okay? Doesn't look like much now because we're just starting. It tells you what the lecture is, what the labs are, what the new assignments are, and what the assignments are due, okay? And so we're going to lecture on chapter 17. You're going to do the lab module. I'm going to show you about Pearson. These are your new assignments. These are assignments due. I actually should have put the student contract in. Notice the student contract is extra credit. It's 1 point. You have 2 weeks to do it, okay? You have a due date here, okay? You have a due date. Notice you have a due date here. This is your roadmap when things are due, okay? Not what is on your Canvas shell. This is your roadmap when things are due. You will get weeks 1 through 12, telling you exactly what we're going to do that week in that class, telling you what the new assignments are, and these old assignments will start stacking up. And I'm going to go over to the other class to show you what I mean by that. So we're in week 4 in that other class. So this was their announcement. So this was their assignments for this week. This is what was due on Monday. Okay. We had chapter 18. We were into chapter 19. They had 2 weeks to do their Pearson and 2 weeks to do their labs. I don't like that because people get behind. So you have 1 week to do your Pearson. We don't have very much on Pearson as far as the homework assignments go. This course is heavily centered on lab, okay, because it's a physiology class. And this shows up, it makes more sense because these are assignments that are due that week. These are the assignments that are due this week, and their exams going to open next week. So I put that there, okay? And so this is what yours will look like starting next week. When we go chapter 18, we have new assignments for chapter 18. Notice I added new. I hadn't did that on the other one. but that's something to be aware of, okay? And so if you tell me that it didn't show up on Canvas, I'm going to tell you that day one of this class, I told you exactly, okay, exactly what you were supposed to be doing. And I put up a week 1 to week 12 or week 15 if it's a 15-week class, telling you exactly what your assignments are and exactly when they're due. If you send me an email, make sure you have read the announcements before you send me an email. Make sure you have read the announcements before you send me an email, okay? Because it's very frustrating for me to spend the time to put these together for you, to make this as seamless as possible, and so that you don't have too many questions. You know exactly what you're supposed to be doing, okay? And go forward. So you get week 1, you get that. These are the usual welcome to email policy. Email policy is very important. You are to use your school email. Do not send me email from your work email. Do not send me a Hotmail. Do not send me a Gmail. Use your school email. My preference is you send it on Outlook. If you send me an email on Outlook, I get it right then. It pops up on my app. I can see it right there. If it's something easy to answer, I can answer it right then, or at least get you started, or at least ask you a question, okay? And if you say, hey, I'm way behind, and I'll say, what are you behind on, okay? Which takes me to the next point. Number 1, now I have 2 classes, okay? I have a Monday class and a Thursday class. So in your subject line, all you need to do, you don't have to write like this email says, VSC 286C. That's all I'm teaching this semester. Just write Thursday. Just write Thursday, okay? Because then I'll know it's your class and not the other class, and I don't have to look through both classes. So in reiteration, you send me an email from the school email. You don't send me an email from your work email. You don't send me an email from Hotmail or Yahoo. You don't send me an email that doesn't have your name associated with it, okay? Which reminds me, when you send me an email, you put your name there. When you send me an email, you put your school name there. You don't have to put your school number. You don't have to put your dual enrollment. You don't have to put what school you're from. I really don't care, okay? All I want to know is what's your name, okay? And what class you're in and what your problem is, so that I can help you with your problem. Usually I respond the same day to your emails. I usually go to bed. I watch either Big Bang, Everybody Loves Raymond, till about 10 o'clock, and then I go to bed, and then I won't be back up till the next morning, okay? So if you send me an email at 11 o'clock, okay, that's not going to help you out, because I'm not going to answer it to the next day. But I will answer it. If I ask you a question, I expect you to answer me back within the same time period. You need to be checking your email twice a day, sometime during the morning, sometime during the evening, okay? You need to be checking your email sometime during the morning, sometime during the evening. If you ask me for help and I ask you some questions so I can help you, and I don't hear from you for 48 or 72 hours, you really didn't need that help, okay? You really didn't need very much help if you can't respond back to me, in a timely fashion, which would mean within 24 hours or 48 hours on the weekend, okay? But if you're asking me something serious, you need to be checking your email, because I'm going to respond back to you, okay? So email policy is very important. Use your school email. This is from Dr. Sterling, who's the head of the Division of Biological Sciences, okay? She's the chair of the Division Department of Biological Sciences. And then we have a chair, Dr. Evans, who's the chair of the Natural Sciences, which covers the biological and physical sciences. So that's email policy. That's very important, okay? Online classes, lectures, and synchronous teaching, which means you should be here at lecture, okay? I get a log of everybody that attends the lecture. If you're one of the smart people who think you're going to log in 4 or 5 hours early, and put it on your computer and just let it be there, and nobody's going to notice that you weren't really there, well, think again, okay? That's not going to happen. It's very important that you're here live if you can. Now, I understand people miss lectures, okay? That's I miss lectures, okay? I miss lectures. I've been to school a lot, so I miss a whole bunch of lectures, and I understand that things come up, okay? But try to be as many lectures as you can. If you're somebody that chronically doesn't come to any lectures, and then you ask me for a whole bunch of help, and I look to see, and you've not been to any lectures, then that's not a sign that you are really very interested in class. You have 2 weeks to sign up on your free membership for Pearson. Okay. How I use that is in 2 weeks, they're going to ask me if you're in this class. If you are not, if you haven't signed up for Pearson in 2 weeks, okay, I would suggest you sign up quicker than that. If you haven't signed up for Pearson quicker than that, okay, and I can't find any record of you being either on Canvas or on Pearson, I'm going to submit your name to be dropped from class. Okay. So you need to be participating in these first 2 weeks so that you don't get dropped from the class, because we have a, it's, if we don't submit that, then the thing goes to the state and then we have to answer why we didn't submit it. And so if I have no record, you didn't sign up for Pearson, you didn't go to Canvas except the first day, okay, you didn't do anything, I'm going to have you dropped from class. So make sure that you participate in class, especially in the first 2 weeks. You need to really participate or you're going to get way behind, okay? So that's the thing about Pearson. We're not going to do, we got into a problem with PhysioX because doing from Pearson and downloading the assignments and uploading the assignments, PhysioX 9.0 is no longer serviced, okay? And Pearson didn't take PhysioX 9.0 off. And so the only way to get to PhysioX previously was to go through 9.0 to 10, because 10 is an HTML file, doesn't require, it was Adobe Flash Player that it was required. So now we do part of our labs directly in Pearson, and we'll talk about that. We do part of our labs directly in Pearson, and we'll talk about that. So we don't have to worry about this so much anymore. We do have to upload some stuff, but we don't have to upload a lot of stuff. Syllabus is here. It's also down in point of information, it's usual standard stuff. If you have the textbook from last year, that's fine. The one thing you do need is a code. Okay, you got to have a code. You got to be on Pearson. You do not go to the end of the semester and say, I didn't know you had to have a code. You have to be on a code. You have to have a code. Okay. So if you don't have one, you got 2 weeks free time to get the code. Okay. You're going to have 4 exams, each 125 points. They are not proctored. Okay. You have 1 week to do the exam. The exam consists of multiple choice, matching, true, false, and short answer. In anatomy and physiology 2, anywhere between 25 and 50% of your test is short answer questions. And we'll have a review before we have the first exam, like I'm doing Monday for this other class. Okay. But you'll have 4 exams, 125 points each. First exam will be on chapters 17, 18, and 19. It's already in the calendar of events. You'll have 1 week to do it. And at the end of the deadline, it automatically submits. So if you didn't start it until 3 minutes before or 5 minutes before it's supposed to be over, and 5 minutes it'll automatically submit, and that's your grade. You have a week to do it. You have a week to do your exam by yourself. Short answer questions, we will go over this, have to be documented where you got the information. Did you get the information from your student notes? Did you get the information from your slides? Did you get the information from the lecture? Did you get the information from the textbook? If you go to Google and Google the answer, and you put a Google answer down there, and I know a Google answer just as soon as I see it, because it has more information than I know, then you'll get a 0. You do that twice, you'll be reported. So it's an open book, open notes test. It's not easy, because a lot of people lose a lot of points on short answers. Okay. It's open book, open notes, but you cannot go for an outside source to answer a question on the test. We'll go over that. So you have exams, 125 points. That makes it 500 points of your score. You have lab modules, okay? Lab modules are just that. They are lab modules, okay? They are, when they're done in Pearson, they're labeled as labs. You have 2 labels in Pearson. We'll look at it. One of them's homework and one of them's labs, okay? The lab modules that are done in Pearson are done in Pearson. They're usually short experiments that you do. You do a pre-experiment quiz. You do the experiment. You do post-experiment quiz, and then use more questions, okay? And then that will be automatically uploaded. Sometimes we have a little bit of a practical or something like that. Nothing like last semester where we had big practicals, and you'll look at some dissection videos and then go to a diagram and fill in the answers. And then that's some of the things you'll have to upload, but I'll explain it as we go along, okay? So you have lab modules. Lab modules, we have a lot of labs in this class this semester. This to me is the harder of the 2 semesters. It has a whole lot of physiology and not much anatomy. A whole lot of physiology and not much anatomy. Last semester was a whole lot of anatomy and not much physiology. We had muscle contraction with the conduction, and we have nerve conduction. That was pretty much the physiology. The rest of it was just anatomy, knowing terms, okay? This is much more complex and much more detailed. So you did a lot of labs, you did a lot of repetition, try to learn this stuff. Counts 300 points. And that's lab. And then we have Pearson assignments, which are homework, okay? They could be as little as a chapter test. For this week, we have Introduction to Pearson. We have chapter test, chapter 17, and we have a thing on blood typing, which, as you can imagine, is very important, okay? And so that's your Pearson homework assignments, okay? That's a category of homework assignments. Sometimes it might just be a chapter test. And so it's not very much stuff, okay? But it is something that you have to do, and you have a week to do it, okay? This is the same calendar of activities you have there. Do not use this as, if the other calendar of activities, if we change the calendar of activities, it will be the one under start of course documents. And it'll say calendar of activities. It'll have that whole name, and it'll say revised, and it'll say when it was revised. This one gets put in when the syllabus gets put in and uploaded to the dean, okay? This one doesn't get changed. So don't use this as your calendar of activities. Use the calendar of activities under start of course documents. So we have 4 exams, 125 points each. Lab modules, 300 points. Pearson assignment, 200 points, for a total of 1,000 points. I do not grade by percentages. Unfortunately, the grading thing comes out as percentages, but it's by points, okay? If you accumulate 900 to 1,000 points, you get an A. If you accumulate 800 to 899 points, you get a B. If you accumulate 700 to 799, you get a C, okay? Most people in this class, the largest majority of this class, get an A or B. You know, that is our goal for this class, for all 24 of you that have signed up for this class to get an A or B, because a lot of you are trying to get into some healthcare career, and you need an A or B on your anatomy and physiology 1 and 2, okay? So that's our goal. I will do everything I can to help you get to that goal, but I can't do the work for you, okay? You have to do the work. You have to stay on track. This is a 12-week class, not a 15-week class. We've got 13 chapters to cover, okay? So there's going to be a lot of work to do. I've tried to space it out as best I can, but there's just no way around it. There is a lot of work to do. That's points, and that just tells you what the grade. You're going to fail if you miss 3 live lectures. We have 12 live lectures. You miss 3 of them, that's 25%, okay? You miss 3 live labs, I call it lecture lab, okay? Miss 2 exams for any reason. You can postpone an exam if you have a valid reason. I need to hear from you. Just don't not take the exam, but I need to hear from you, and I will schedule the makeup at the end of the semester, which is really not when you want to be taking an exam. If you miss 2 exams for any reason short of being in the hospital and on a ventilator, okay? There's something that grave, because exams don't come that close together, then you're going to fail, okay? You have a week to take the exam. You have a week to take the exam. So sometime during that week, you can take the exam, okay? You just have to, the exams are 2 hours long. You get your computer, you sit down in front of your computer, you get you something to drink, and you start. In 2 hours, it's going to be submitted. You can get up and go to the bathroom if you want to. You can have your book by you. I told you, open book, open notes. But you need to take the exam. Incomplete grades, there's a protocol for incomplete grades. You usually have to complete 75 to 90% of your work, be passing. Okay, so if you only completed 25% of your work, you're not going to be able to be an incomplete. Okay, you have to complete the majority of your work and be passing, and I have to approve it, and we have to sign a contract, and you have to make it up the next semester, and you get an I until you make it up. Okay, so if you're trying to get into a program, it's not going to go well for you if you got an incomplete, because unless you intend to make it up early on. Make up work. You can miss one exam. You need to let me know if you're going to miss an exam, and let me know. Have a valid reason to miss it. Okay. But just let me know. You're sick. Your family's sick. You're going to be traveling. Work's killing you. Just have a valid reason and let me know what it is. Don't send it to me after the exam closes. Okay. Lab modules. Okay. There is a due date on everything. We're not talking about exams. On exams, there's a due date. When the due time comes in your class at 6 o'clock p.m., when the due time comes, it will be automatically submitted. Okay. Lab modules, there is a due time. Okay. If you want to earn full credit, on your lab module, you have to submit it by that due date. For every day late after that, it's 2% off up until 10 days, at which point it would be 20% off. Okay. So if it was 20 points, okay, if it was a 20-point lab module and you submitted it, day 12, you would get 4 points off, you get 16. Okay. Well, that's 20%. So if you do that to all your lab modules and all your Pearson assignments, the same thing. 2% a day for every day late it is, up to 10 days late, and then you get 20% off. After that, if you want to wait till the end of the semester to do them all, that's fine with me. You're still going to get the 20% off, and 20% of 300 is 60, and 20% of 200 is 40. So 60 plus 40 is 100. You've already lost your A. And I did this because I let people do assignments up until the end of the semester, last semester. And a lot of people waited to the end of the semester. They got no benefit from the assignments. And I'm not going to do that again. And it wasn't fair to the people who did their assignments on time. So yeah, you can turn in a late assignment, you 2% a day, each day for 10 days up until a maximum of 20%. You can turn it in up until the last day of class, but you're still going to get the 20% off. So make sure you turn in your lab modules on time, and make sure you're turning in your Pearson assignments on time. Pearson assignments, there's general, are due 1 week later, unless we have, the only holiday we have during this class is spring break. So if we do a chapter the Thursday before spring break, obviously that Pearson assignment's not going to be due that next Thursday, because we're on spring break, it'll be due the following Thursday. Okay. Your expectation for student contact. Number one, if you need to get in the study group, get in the study group. If the study group's going to be productive, it's not going to be productive, don't get in it. Don't let yourself get too far behind before you ask for help. I talk a little bit harsh and rough, but Dr. Sterling will tell you there's never been a student I didn't try to help. There's never been a student I didn't try to help. So if you get behind, let me know so we can get you back on the road to getting you your A or B, okay? And getting you into your program or whatever you want to do. I notice we have a lot of people doing this for personal objectives. And so a lot of this is just standard stuff on here. And so now we've gone over the syllabus. Let's see, where had Pearson at? All righty, here we go. We'll do it again. All right, we're going to Pearson. Here's your class, BSC 286C. I don't know why I didn't capitalize that. I'm just making the class up. Down here, structure tools, go down here where I click assignments, where I assign assignments. Okay, these are your assignments, okay? Your assignments for Pearson homework, notice it says homework here, is Introduction to Mastering A&P. It is the Chapter 17 test and Chapter 17 blood typing. You'll just go through that exercise once you finish it. You know, my philosophy on these is you go through them up until the deadline, okay? Up until the deadline. Then you'll have a lab module. Notice that these are due next Thursday at 6 p.m. And your lab module, remember the Pearson's going to be doing 1 week, the lab module is going to be doing 2 weeks. How do you know? You go to your whatever week 1, week 2, week 3, you go to your calendar of events. It says that on your calendar of events. This is your lab you're going to go to. It's called blood typing, okay? You can go to this lab, you can go to student, well, you're already student view, you're going to do this. Go here, it's going to have some questions for you to answer. It's going to have an exercise to do. You'll do whatever this says to do. It'll have some reading for you to do, okay? You'll do the reading and go through the reading. It'll tell you about the equipment. It'll tell you what you need to do. It'll have some questions for you to answer. And then it'll have you do the experiment, okay? And it'll tell you exactly what you need to do. Drag a blood typing slide from the slide to the workbench, okay? So it'll tell you what to do, to drag a slide from the slide to the workbench, okay? So it's not hard to do. These things are not hard to do. This is what we used to do through PhysioX, which was the same thing, except now since Pearson didn't pick up, even though Adobe, according to one of my other students, warned them that a year in advance that 9.0 Flash Player was going out. We're not able to do it, but we can do it here. It's the same experiment. Okay. So you're going to have 2 exercises in blood typing because blood typing is so very important. Okay, it's so very important. So that's how you do those. Eventually, you'll get to the end, and they'll ask you to submit, and just submit it, and it automatically gets graded, and your grade goes there. Okay. So, and I will tell you, we'll get back to the modules. Okay. But this is Pearson. Those are your assignments. Okay. We looked at the assignments. So for this week, you have the introduction. You're going to have a blood typing assignment and a chapter test. And then you're also going to have a lab module, which is blood typing. A lot of times you're just going to have a chapter test. Okay. A lot of times you're just going to have a chapter test unless we have some of these things like gas exchange when we get to the respiratory system or how the blood flows through the heart. But most of the time you'll just have a chapter test as your homework. Okay. So it's not that it's too grueling to do. And so you have a week to do that, and you should be able to get that. You know how to register that, and so that should be okay. Let's get back to here's your registration, back to our class here. So your student contract, this is your student contract here. You'll download your student contract. Okay, you'll sign your student contract, date it, and put the course as semester. Okay, you will then upload your student contract over here to assignments. You go to assignments. It says student contract, extra credit. Okay, you'll upload your student contract here. And you'll upload it. This is your assignment. Tells you what to do. Okay, yours will look different. This is mine. Okay, but it tells you what to do, what to name it. Okay, and how to upload it, and you'll upload it, and that'll be the end of it. You got 2 weeks to do that. You're going to have to do it before you get a grade. You're going to have to do your student contract before you do a grade. You might as well do it in the first 2 weeks and get 1 point of extra credit. You might as well do it in the first 2 weeks to get 1 point of extra credit. The second assignment is the discussions here, okay? Close for comments. I'm going to open that up, okay? But you'll have a discussion board, okay? You have a cyber cafe here. Cyber cafe is for you to talk to other students. You don't have to use it. It's not something I monitor. It's only about class stuff. Hey, what did he say about this? Or do you want to form a study group? You can use it if you want to. Some people use other apps, okay? But you can do that. But I'll get the discussion board set up so that what you're going to do is put 2 paragraphs. The first paragraph is a little bit about you. Second paragraph is what you hope to achieve. Hey, I'm freshman FSCJ. I'm going into nursing. And a couple of maybe 3 or 4 sentences each time. Okay. But it's not anything very elaborate. You don't have to put in any information you don't want to. You don't have to do it if you don't want to. Okay. But it's 2 points of extra credit. And since we don't have face-to-face meetings, and you can't say, be sitting next to somebody and find out, hey, they're going into physical therapy too. And so we can share notes. Okay. We can share notes about what's going to go on. You don't have that benefit because, so this gives you a way to network with other individuals, because I'll tell you, you get your first job based on your schooling. You get every other job after that uses some network. I got this job because of network. Ken, who I can't remember, I met him. I was teaching physics in high school. And I went to a Florida Physics Society meeting in Gainesville. I met him and Ken Schechter. He was teaching engineering. And I said, what are you teaching? What else you do? I teach the FSCJ. I said, I'd like to teach at FSCJ. He said, okay, I'll talk to the dean. Talked to the dean. Dean said, send your CV. I sent my CV, and I got a job. Okay. So networking is very important. Never, never, never discount networking. Okay. Never discount networking because it's very, very, very important. So let's go back here to home. Okay. So modules here. This is our first module. This tells you how to do the blood thing. Okay. And so you get instructions. So every live module will tell you what to do. Even gives you example of the 2% off a day. Okay. Worth 15 points. You have until Thursday at 6 o'clock on this day to complete it. Okay. 2% deduction for that. This is what the module is called. It says lab there. Okay. So it'll be segregated out from your homework. Okay. Remember, lab counts total points of 300 points at the end of the semester. Pearson assignments, all the Pearson assignments count 200 points. So you'll get something every week to tell you what to do, so you won't be in the dark about what to do here. Okay. At least I hope you won't be in the dark about what to do. You have PowerPoints, you have student notes. These in the other class were filling the notes. It looks like here I must have put, no, but these are filling the notes. Okay. So these are going to be your bases. These are going to be your bases for when you either follow the lectures, listen to the lectures, whatever, fill these in. Okay. These are going to be your basis for studying. Okay. You won't get a 90 just by these notes, but you probably if you know and understand these notes, okay, remember you're going to have these notes available to you, can get an 80, okay? But you at least a 70 to 80. And then if you know the notes, the slides, and listen to the lecture, because I will literally tell you some of your questions that are going to be your short answer questions, or I'll tell you what's important, okay, as we go along. But if you don't attend class and you don't listen to the lecture, because it doesn't matter if it's the lecture from last summer, from 2 semesters ago, because these exams, most of the exams, I revise a little bit, but the important things are still the important things. Okay. So these are your notes. You need to fill them in. Okay. Fill them in and let this be the basis of what's important to you, to study for your exams. You'll have 3 sets of notes for each one for each of the exams. Okay. And you have PowerPoints. Okay. Here's the PowerPoints we're going to use. This recorded lecture, I still haven't got it off Zoom. I'm going to try to get it off of Zoom. This lecture actually comes from YouTube. So this guy is good. Okay. If you ever want to get additional information off of YouTube, there are a ton of lectures out there. Just type in Merib, who is the author of our book. Okay. And write chapter 17 blood, or just type in blood. All these lectures come out. Okay. All these lectures will start pouring out from different instructors who have put them up on YouTube. Some of them are high school anatomy and physiology because they have AP anatomy and physiology, but they're good. When I started teaching 5 years ago, this is what I watched because I was a plastic surgeon. So you think I remembered much renal physiology? No. Okay. Think I remembered much heart physiology? No. So I ended up watching a whole bunch of these lectures. Okay. So if you ever want to see a lecture, okay, if you ever want to see a lecture, just type in Merib, who's the author of our textbook, and whatever you're trying to look at, you could just type in Merib blood, okay, and it would take you to all the lectures from the Merib that they use the Merib textbook, and the Merib textbook is the most common, most used textbook in the country, okay, for anatomy and physiology, both at the high school and at the college level. Okay, it's a very good textbook. I would suggest if you bought the textbook, you keep the textbook. Okay. I would suggest if you bought the textbook, you keep the textbook because if you ever rotate onto a surgical service, you're going to want to review the anatomy before you go into the old operating room the next day. I can tell you that from a fact. Okay. Tip number 2. Tip number 1 was networking. Tip number 2, if you are on a service doing your internship, rotation, fellowship, whatever they call it, and your mentor asks you a question, you don't know the answer, that's perfectly fine. There's a lot of stuff you're not going to know because there's a ton of material you have to learn. But what I did when I was a resident and chief resident and a fellow, okay, if I asked a medical student a question and they didn't know it, that was okay. But the next day I asked them the same question. And if you don't know it that day, that shows you're not paying attention and don't really care. Okay. So remember that if somebody asks you a question, it's not dishonorable to not know the answer to the question. It is dishonorable and shows a lack of interest if you don't go look that question up that night. If you don't go look that question up that night. Because I can tell you when I was a medical student, I got asked tons of questions, absolute tons of questions. Okay. And didn't know most of the answers, but after a while I learned to look them up. Okay. So we have that. We have these 2 recorded lectures, and this is a blood typing worksheet. It's not really a worksheet. Okay. Blood typing is very important in this chapter. You're going to have a bunch of questions on blood typing. And so I made out this sheet. Okay. It tells you the antigens on the red blood cells, tells you the antibodies on the red blood cells. Excuse me, it tells you the antibodies in the plasma. It tells you what blood people can get, okay, what the universal donor is and what the universal recipient is. So you're going to want to be able to work through this sheet. Ideally, you'd want to be able to, if you had this blank sheet, and you won't have this on the test, okay? You won't have to fill in a blank sheet. You won't have that. But if we're doing a face-to-face meeting, you might, because in the lab, I might, or as an extra credit question, I might give you this table with nothing filled in and you fill it in. But this is your roadmap because you have 2 activities on blood typing. You have one in Pearson homework and you have one in the Pearson lab module. Okay. And so it must be very important. Okay. Must be very important. So it's something you're going to have to learn. I don't know why that bumped up. And like I say, we have a bunch of lab modules. And so this is 18 cardiovascular, pretty well same thing. The PowerPoints, the introduction to the notes. See, that's why I hate that. It just takes too long. And here's cardiology with notes to be filled in. Okay. But it goes along the same. It goes along the same way. I will open this up. I'll usually on Sunday. Okay, for you. The rest of these are closed. I only open up this. You're free to start working. Okay, if you want to. You'll have the week's assignments. You'll have the notes. You'll have the student notes. We just did on Monday. We did blood vessels in the other class. Okay. And so this is what they're working on now. These are the two activities they're working on in Pearson: the lymphatic system, the immune system, respiratory system, digestive system, urinary system. Once we get through the urinary system labs, there won't be this many labs because we can't do the Physio X now, so I'm having to do more stuff on Pearson, but I'm revising them as I go in the other class because they're a couple weeks, 3 weeks ahead of you. And then once we get past that, there's not really any more labs after that, except for this one genetics lab module. And it's just a little short handout to do to help you with genetics. And so that's how the course is set up. Okay. Don't worry if you don't understand everything. All you need to do is start working on chapter 17. You have the student notes, you have the PowerPoints, you have this recorded lecture. Okay. And you have this worksheet and you have your assignments. And so we're going to talk about the PowerPoints for a little while, okay? We won't get through them all. We're going to talk about them for a little while. And so this is blood. These are Dr. Oler's PowerPoints. In fact, when we get about halfway through the class, the recorded lectures, not the ones that I did, but the recorded lectures, actually her lectures. Because if you were in class March a year ago, or it'd be a year, you're in class then, we transitioned literally over spring break, over 2 weeks. We went from face-to-face meetings to online meetings. And so she was my mentor. She's actually the head of anatomy and physiology and a great person. She gave me her slides, her student notes, and she gave me her recorded lectures going forward. So about once we get to it, and I'll let you know, that's actually her recorded lecture, so you can listen to what she has to say about it. We're going to talk a little bit about blood, okay? In our first unit, we talk about the medium, which is blood. We talk about what pumps the blood around, which is the heart. And we talk about the highways where the blood is pumped, which are the blood vessels. So that's your first block module, and it's all related, okay? It's all related about what we talk about. Functions of the blood. A lot of this I'll read directly from the slides, and I know it's a little bit boring, okay? But there's really not a whole lot to add, okay? And you've got those notes there that you can work with, okay? And they'll be available to you. Like I say, I'll try to get them up on Sunday so that you can download them, fill them in if you want to. Whatever you want to do. You don't have to do anything if you don't want to with them. They are for you. They are the basis of your studying for your exam. So I would download them. I would fill them in, and then as Dr. Phillips said something extra, I'd put it in. If he said that was important, I would star it, okay? If he says it's going to be on the test, I would big start or double start or triple start or whatever you're going to do, okay? But there are ways for you to help yourself, and I will give you hints as we go along. Distribution, delivery of oxygen to the tissues and CO2 to the lungs. Every cell in our body requires O2. Every cell in our body does aerobic respiration unless you're getting into an anaerobic state if you're doing a lot of workouts or things like that. But in aerobic respiration, you're taking glucose in the presence of oxygen, you make CO2, water, and ATP. Okay, that CO2 has to be removed from the cells and taken back to the lungs. Well, how's it going to be do that? It's going to be transported in the blood. And so one of the questions you're going to see on the exam is how's O2 and CO2 transported in the blood? And I guarantee you at least one of you is going to write O2 and CO2 is transported in the blood. And that's probably a 10-point question, and you're going to get 1 point for lead. I had a finance professor when I got my MBA. he gave you 2 points for lead. And so you probably, if you wrote a little bit more, you'd probably get 2 points for lead and you'd lose out 8 points, okay? And we won't talk about that now. We'll talk about that obviously when we get to the lungs. Deliver nutrients from the digestive tract to the cells, right? Take metabolic waste from the cells, deliver hormones to endocrine organs or target organs. Regulation, body heat. If you go outside and it's hot, you can look at your arms and the veins have dilated up. Why have they dilated up? They are trying to keep your body from overheating, from getting heat stroke or heat exhaustion and heat stroke, okay? So they are trying to dilate up. They're dilating up to dissipate the heat. Now, the problem is if it's 100 degrees outside, it's hard to dissipate heat into that hot environment. That's why you hear about a lot of older people dying in St. Louis and Chicago during those heat waves, and all they've got is fans because all they're blowing on themselves is 100-degree heat. And so they're not able to get rid of their body heat. Okay, maintains pH by way of buffers. Fluid volume, to keep the volume in the blood vessels, we need volume. Protection, prevents blood loss. Prevent infection, that's how we carry our cells that are prep for infection through the blood. Compositions, it's 8% of the body weight. It is a connective tissue. If you remember from our Anatomy and Physiology 1, blood was a connective tissue. Remember, connective tissue was the hardest of all the tissues, okay? 5 to 6 liters for males, 4 to 5 liters for females, and that's only a relationship of size, okay? If you have the same size male and same size female, okay, you'd have the same amount of blood. It's only because, as a general rule, females are smaller in stature and way less than males, and that's why you have differences in volume. Normal pH of blood, 7.35 to 7.45. Notice it's not 7, it's not neutral. It's actually a little bit alkalinic, okay? It's on the alkaline side. You might want to star that. That's going to be a fact. It's probably matching a multiple choice or true false, okay? Components, connective tissue. You have plasma, okay? Blood, the liquid part of the blood is called plasma, okay? You have formed elements, the cells. Plasma's non-cellular component makes up 55% of the total blood volume. What's the difference between plasma and serum? If you draw blood and put it in a tube and it clots, you're still going to have a fluid above, okay, especially if you spin it, or if you leave it there, it'll settle out. You'll have fluid above and you'll have the formed elements of cells below. What's the difference between plasma and serum? Plasma still has the blood clotting factors. They have not been activated. Serum no longer has the blood clotting factors because the blood has clotted, okay? Formed elements, we've got all kinds of cells. We've got some red blood cells here, okay? The reason they're clear in the middle is not because they're a donut, but they have a biconcave shape. So that middle part is actually indented on the front and the back. We have leukocytes. These are the white blood cells here. And we have platelets, which are these little fragments here, which are available for clotting, okay? Composition, so these are our red cells here, these are our platelets here, and these are different types of white cells. Plasma, 90% water. Remember, we're made solids. We have in it's albumin. That's our #1 protein, okay? pH buffer and major transport protein contributes to plasma osmotic pressure. The number of dissolved particles in the fluid results in osmotic pressure. We have globulins, alpha and beta. We have gamma globulin for antibodies. We have fibrinogen, which is a clotting protein. We have waste. We have nutrients. We have respiratory gases. We have electrolytes. If you ever have blood work done and they draw sodium and you have a chemistry panel and they do sodium, potassium, and chloride and phosphate, those are some of your electrolytes. And hormones are there. Serum, plasma without fibrinogen, in other words, without clotting factors. Erythrocytes, the most numerous cell type. Females, 4.2 to 5.4 million per millimeter cube. That's also cc, or also milliliter. Males, 4.7 to 6.1 million per millimeter cubed. Hematocrit, total blood volume occupied by the red blood cells. If you spin the blood down, okay, if you spin the blood down, you'll have a certain part of it that's fluid and a certain part of it that's red blood cells, okay? The white blood cells are just what's called a buffy coat. It's a white layer on top of the red blood cells. Well, if you measure that, if the total volume is 100%, in females, 42% of the volume would be red blood cells, and 47% of the volume in males would be red blood cells. This is a shape. This is a biconcave shape. The reason it's like this is red blood cells, they really have one function. They have one function, and that's it. They carry hemoglobin. Oxygen is attached to hemoglobin, okay? So this gives them more surface area so they can absorb hemoglobin, okay? Gives them more surface area so they can absorb hemoglobin. And when it gets down to the cellular level so the hemoglobin can be detached, they have no nucleus. They have jettisoned in their process of development every non-essential organelle. They have jettisoned every non-essential organelle, okay? So that they can maximize the hemoglobin capacity, hemoglobin carrying capacity. And they got a shape. This is some of the things that are in there, okay? They live for about 120 days because you can imagine they can't reproduce. They can no longer have a nucleus. They've jettisoned the nucleus. Function transport of respiratory gases, oxygen transported by hemoglobin. As oxyhemoglobin, where oxygen's attached. If oxygen's not attached, it's called deoxyhemoglobin. Just like you had ribonucleic acid and deoxyribonucleic acid. You have oxyhemoglobin and deoxyhemoglobin. All carbon dioxide transport is primarily bound to the globulin, okay? When the O2 is not attached, forms carbaminohemoglobin. I can never say that word, okay? If it's attached, however, CO2, the majority of it doesn't travel by way of being attached to hemoglobin. Blood cell physiology, synthesis of hemoglobin, 97% of the dry weight of the red blood cell is hemoglobin. So you take out the water, it has hemoglobin there. Normal amounts, when you measure hemoglobin concentrations, this is what it is in males and this is what it is in females. If you had same size males with same weight, you'd have the same value, okay? Site of formation, immature red blood cells are in the bone marrow. They continue for a short time and then they're released to the circulation. So they mature in the bone marrow. Don't forget that hemoglobin is 2 alpha and 2 beta protein chains. This is our example of a quaternary structure in proteins. This is hemoglobin, okay? And we have an iron molecule in each one of the hemoglobins. Iron-containing components synthesized in mitochondria and variations on the Krebs cycle. Globulin produced in the ribosomes. There are 2 alpha and 2 beta chains. Each contains about 120 amino acid units. One heme is associated with each chain. So you have one chain, each of the one chains has a heme, and heme. Each iron and hemoglobin can bind with an oxygen molecule. Therefore, each hemoglobin molecule can transport 4 oxygen molecules if fully saturated. Majority of the time it's fully saturated or almost fully saturated until we get to the tissues where it's offloaded. Each red blood cell contains about 250 molecules of hemoglobin. Each hemoglobin has 4 chains with an iron, can transport 4. So we can transport about 1 billion molecules of oxygen, okay, in each red blood cell. And we need it all. They generate ATP aerobically. They got no mitochondria, okay? And they don't use the oxygen they transport it, okay? So everything was made for them as they matured. Erythropoiesis is the development of the red blood cells. Many sites in the fetus, an embryo, liver, lymph nodes, etc. Later primary sites, red bone marrow of the axial skeleton and the pelvis. Okay, hemocytoblast, that is your primitive cell. You don't have to know this. You're not going to be asked what are each of the individual stages here. This is not a hematology class. There's not a blood class. This is just a survey, a chapter of the blood. But the progenitor cell, the stem cell, is a hemocytoblast. So you might need to know that it's a stem cell. Notice here, as we get to the normal blast, remember I told you the nucleus was jettisoned, okay? So the nucleus is jettisoned before it gets released out here. An early red blood cell is called a reticulocyte. If you do a special stain, you'll see a few of those in the peripheral blood, but you shouldn't see too many, and that's a mature red blood cell. Proerythrocytes synthesize hemoglobin. Okay, so you get the proerythrocyte, and so you're starting to synthesize, proerythroblast are starting to synthesize hemoglobin. Later, the nucleus is expelled, forming the reticulocyte stage. Takes about 15 days, and they're released to the blood. Okay, see, there's my picture there. I don't look that good. Okay, I may have looked that good at one time. Right now, I have long hair. My glasses are taped up in the middle because I broke them. I had to go see the eye doctor to get some new glasses. Control of erythrogenesis is dependent on dietary factors and hormones, regulated back by a negative feedback system. We know a negative feedback system. Glucose had a negative feedback system. Glucose got high, you manufactured insulin, okay, to drive the glucose into the cells. If it got low, you manufactured glucagon to mobilize glucose to get it into the bloodstream. That's a negative feedback system, okay? Too little, it causes hypoxia. Too much, the blood's too viscous. Materials required. You got to have B12. You got to have an intrinsic factor, which was made in the stomach. We'll learn about that. We'll learn ad nauseum about the physiology of the stomach and the GI system. Stored in complexes called ferritin, transported by transferrin and hemosiderin. Hormonal control. Decrease in tissue oxygenation causes increased release of erythropoietin in the kidney. This leads to increased maturation of red blood cells in the red blood cell pathway. Used to some Olympic athletes and endurance athletes would do blood doping. Blood doping is giving yourself some red blood cell infusions, okay, to give you more red blood cells so you can carry more oxygen so you could compete at a higher level. Then they got to testing for blood doping, so they started taking erythropoietin, and now the Olympics test for erythropoietin, EPO. Okay, erythropoietin is something that you can give to somebody to increase their red blood cell count, okay? If it's a chronic anemia and you don't have to increase it fast, that's something you can give. If you ever have a Jehovah's Witness patient, okay, that won't take blood, then they have a special number to call, and they have a special erythropoietin to give them that's not blood-based. That I don't. Let me rephrase that. I'll retract that last statement. A special erythropoietin to give them to increase their blood production. Now, if they're in a trauma situation, that's a different situation. Or if they're actively losing blood, then you have to talk to them about a different type of situation because erythropoietin is not going to act that quickly. But it is something that causes increased red blood cell production. Reticulocyte count normal 1 to 2. An increase indicates good response to blood loss. A decrease is a shortage of erythropoietin, our inability to respond to it. Normal rate is 2 million new cells per second in healthy people. Destruction, necessity for destruction, limited physiological capabilities. Okay. It can't do anything. It doesn't have a nucleus. It can't undergo mitosis and reproduce itself. It only has the ability to carry hemoglobin, does glycolysis by anaerobic respiration, so it doesn't produce very much ATP, only what it needs to do, doesn't use the oxygen that it carries, maintains minimal functions, squeezes itself through some very tight places, because when you get down the capillary bed, you're talking about very, very tight places where the red blood cell actually has to squeeze itself to a smaller and smaller shape, longer and longer shape, okay? So they're not going to last very long, okay? Wear and tear, numerous strips of the circulatory system, they get fragile. They're going to break down in about 100 to 120 days. Tissue, macrophage system, phagocytosis, spleen, circulatory channels, liver and bone marrow. We'll discuss this a little more when we get to the lymphatic system and the immune system, but they're primarily broken down in the spleen, okay? Hemoglobin is going to be broken down. It's going to be broken down into heme and globin. Okay. Everything that can be used will be used. Okay. So the globin's broken down into amino acids and recycled. Okay. Just like when we eat animal protein, we can't use animal protein, but we can break that animal protein down into amino acids and utilize those amino acids to build proteins that we can utilize. Okay. Because animals have the same amino acids we do. Iron is released and transported, may be stored in the liver. Okay. If it's broken, it can be resynthesized, or if you don't need that iron, that's going to be broken down to biliverdin and bilirubin. It's going to be absorbed by the liver, conjugated, okay, then going to be secreted, and it's going to come out as bile. That's responsible for some of the brownish stool coloring, okay. In the intestine, bacteria converts it into urobilinogen, which is a pale yellow. It can then be reabsorbed and filtered out by the kidneys and secreted in the urine. Some remains in the intestine, undergoing further transformation. So the iron part, everything else is broken down. The iron part is either reutilized in resynthesis of hemoglobin, or is excreted by way of the intestine or the kidneys. Okay. Jaundice occurs when excessive bilirubin is formed in extracellular fluid. Common causes are blockage of the bile duct, because the bile comes out of the liver and stored in the gallbladder. You get the bile duct, which is the connection of the gallbladder to the common duct, which is going to drain into the intestine. You get a blockage there. You can get liver disease, cirrhosis, or you get hepatitis. Red blood cell disorders are anemia, lacking blood, low oxygen carrying capacity, usually of symptom. Okay. Insufficient numbers, you can have hemorrhage. Okay. Blood loss can be acute or chronic. Acute could be a trauma situation. Chronic can be a bleeding ulcer. You get hemolytic from transfusion reactions or from bacteria or parasites. You can get iron deficiency anemia. You can get a genetic disease called thalassemia. You can get pernicious anemia. You get deficiency of the intrinsic factor, which is not absorbed in the stomach. Or if you've done a partial gastrectomy or near partial or total gastrectomy, then they're not going to be able to absorb the intrinsic factor. You can get sickle cell disease, which causes the cells to be abnormal. See, normally this is a red blood cell. If you get sickle cell disease, they sickle. Okay. What happens now, instead of being able to squeeze, and remember, this is just a bag of hemoglobin, so you get into a narrow space, it can just it's like taking a balloon that's partially filled with water. You can squeeze it and put it through a hole. Okay. Well, if the balloon is rigid, if that water's frozen, you're not going to be able to squeeze it and get it through the hole. This is what happens with the sickle cell. Okay. So if they get into a sickle cell crisis, they get a lot of pain, because a lot of these are blocking a lot of small blood vessels. Okay. And it can eventually result in hypoxia and necrosis. Polycythemia is excess red blood cells, highly viscous blood, bone marrow cancer, or dehydration. That's not a good thing, because it affects the blood viscosity. Secondary polycythemia, high altitudes. If you're somebody that lives at high altitudes, you'd have your red blood count and your hemoglobin all be much higher. Okay, because remember, the higher up you go, the less oxygen there is. Okay, the less oxygen there is. Increased work of the heart, because the blood's thicker, can increase risk of stroke. Okay, we got leukocytes, normal, 4,800 to the way I remember, it's 4,000 to 11,000. That was my, a lot of this is ballpark stuff. So when you're up on the wards, I remembered 4,000 to 11,000. Every lab is different. That's why they give you a normal value when you get a lab report, because every lab is different. I mean, it won't be 3,000 to 15,000. It won't be that much, but it might be 4,500 to 10,500 or something like that. So every one of them's different. Okay. They have nucleus. They have no hemoglobin. Use diapedesis. Diapedesis is squeezing. Okay. Squeezing between the gaps to leave the capillaries. Types and subdivisions. We have granulocytes. We have 3 types of granulocytes. We have a neutrophil, an eosinophil, and a basophil. Neutrophil has neutral granules. eosinophil has red granules. A basophil has dark blue granules. Okay, these are the three granulocytes. These are the two agranulocytes. This is the lymphocyte, the smallest of the cells. And this is a monocyte, which has a multilobulated nucleus. And so one of the things that we used to look at was peripheral smears on the blood when we did the face-to-face labs. Neutrophils, by and large, they're the highest percentage, 50 to 70%. This is a neutrophil with neutral granules. These are red blood cells. Normal numbers 3 to 7,000. Function: phagocytized bacteria make defensins and lysosomes. Chemotaxis attracted to area of inflammation. So if you have an area of inflammation, you have a cut, you have something like that, the neutrophils are going to be attracted to that area, okay? Because either the damaged cells or if it's gone longer by the microbes, and they're going to be attracted. If you have a greater increase in neutrophils, either by way of percentage or by way of numbers, it usually indicates a bacterial infection. These are eosinophils. Normal number 100 to 400 per cubic millimeter. Notice that the neutrophils were like 3 to 7,000. These are 100 to 400 per cubic millimeter. You may go for, if you work in a lab, you may go for weeks and never see one of these, okay? Function: attack parasitic worms, lessen the severity of allergies by phagocytizing antigen antibody complexes and inactivating the inflammatory reaction. Basophils. This is a basophil. Normal numbers 20 to 50 per cubic millimeter. I worked in a lab better part of a year, or a couple of labs better part of a year. I rarely, if ever, saw an eosinophil, because once you see one, I mean, they got those bright red granules. They stick out like a thing. I never saw a basophil because their numbers, you have to remember, normal number of white blood cells is 4,000 to 7 to 11,000 ballpark. They per cubic millimeter. They are in ratio of 20 to 50. And I don't ever recall seeing one, unless it was a prepared slide where somebody had increased basophils, and they, you were looking at it on a slide like in medical school. Release histamine and heparin, which intensifies the inflammatory response. Differentials increased in chickenpox, sinusitis, and diabetes. Okay, so neutrophils, infection; eosinophils, parasitic worms; basophils, chickenpox, sinusitis, diabetes. Agranulocytes, no staining granules. These were our granulocytes. There's 3 of them. These are agranulocytes, more regularly shaped, okay, nuclei. The lymphocyte, the nucleus is primary, all the cell. And then the monocyte, the nucleus is multilobulated. What differentiates that from here and here is the fact that this has granules, okay, and this does not have granules. And it's usually bigger, okay? Lymphocytes, 25% of that 4 to 11,000. 3 sizes, small, medium, and large. I never differentiated that. Normal number 1,500 to 3,000. So of the 4 to 11,000, 1,500 to 3,000 are lymphocytes, mostly live in lymph tissue. They monitor the lymph tissue as it comes through, looking to attack stuff, okay? Non-self stuff. And when we get to the immune system, we'll learn about self and non-self. So if it's non-self stuff, they're looking to attack that. By direct attack, T cells are antibodies, so they can either attack it directly. There's actually 2 different types of these, and they can attack it directly or they can make antibodies to it. Differential increase in acute viral infection or immunodeficiency. Monocytes, 3 to 8%. This is a monocyte here. Multilobulated nucleus, usually bigger. 100 to 700, you'll see on peripheral smear, usually see a monocyte. Phagocytosis after differentiation into a macrophage. So this turns into a macrophage, which is the scavenger of the white blood cells, and goes out and eats stuff, okay? I know that's not a medical technical term. It goes out and ingests stuff, okay? And if it finds a foreign antigen, think injection of the COVID virus vaccine, finds a foreign antigen, it will ingest it and it will present that antigen to the immune cells to start a response. Differential increases with virus, intercellular bacteria, chronic infections, production of white blood cells, Hemocytoblast is also a precursor cell, then becomes either a lymphoid stem cell or myeloid stem cell, turns into myeloblast or lymphoblast. So these are, there again, you just need to know the names. You're never going to have to do this, not in this class, okay? You're going to have to know these cells, you're going to have to know the granulocytes and the agranulocytes, but you're not going to have to be able to go down through this hierarchy here, okay? Lymphocytes, lymphoblasts become lymphocytes, myeloblasts constrict nucleus and become band cells, accumulate lysosomes, become granular leukocytes. One type also becomes a monocyte. So the lymphoblast only makes lymphocytes. The myeloblast, okay, makes all the granulocytes and the monocyte. That could be a trick question, okay? I don't think something, but it could be a trick question. Leukocytosis, high number above 10,000. Leukopenia, low number above 5,000. There again, based on your reference lab. Leukemia, immature white blood cells flood bloodstream. Bone marrow dominated by cancer. So what happens is the healthy cells get crowded out. And so instead of looking at a peripheral smear with all those mature cells, okay, you're going to look at a peripheral smear and see all those immature cells, okay? And that's going to be your first hint that it's leukemia. Platelets, normal number 150 to 400,000 per microliter. That's a micron, that's 10 to the minus 6. Thrombopoietin regulates formation of the megakaryocytes, starts as hemocytoblast, okay? And then goes, notice that platelets are literally just a fragment of the cell. They break off, these are the platelets. They're just fragments of cells, okay? So they're fragments of cells. They're responsible for clotting, break off into the circulatory system, blood clotting, hemostasis and blood clotting to stop bleeding, 3 stages. We have vascular spasm of the smooth muscle and the damaged vessels contract, decreasing flow from the site of injury. So the vessel first goes into spasm unless the vessel is too big to go into spasm. So it's femoral artery, it's not going to spasm. Can last several minutes to half an hour. Platelet plug forms. So you're going to have spasm, then a platelet plug forms. Platelet adhesion causes it to adhere. It releases, it's going to release factors, okay? It's going to enhance vasoconstriction and increase stickiness of other platelets. And then you're going to have coagulation, the clotting mechanism kick in. You're going to have prothrombin, which is going to be activated, okay? So this is you have open book, open notes, but this is the basic clotting mechanism, okay? You have prothrombin activator, it converts prothrombin to thrombin, and that's going to convert fibrinogen to fibrins. That's you have 2 pathways, an extrinsic pathway and an intrinsic pathway. These, you're not going to have to know these, you just need to know there's 2 pathways, okay? The extrinsic pathway is triggered by the release of chemicals, tissue thromboplastins from traumatized tissue, comes from outside the blood vessels, is extrinsic, requires vitamin K and calcium. It is very fast. So extrinsic is outside, comes from release of chemicals. It requires vitamin K and calcium and very fast. Okay, intrinsic pathway, triggered by platelet activity. When they come in contact with this, requires vitamin K and calcium, also clotting factors. It's very slow. So it's triggered inside the vessel, the other's triggered outside the vessel. And so that's your big take-home message, okay? Extrinsic, outside the vessel, requires vitamin K and calcium, very fast. Intrinsic, inside the vessel, requires vitamin K and calcium, very slow. And remember what the activators, but there again, this open book, open notes test, so it's not as hard. This is our prothrombin. Prothrombin goes to thrombin, okay? And eventually you're going to have fibrinogen and converted to fibrin, and this fibrin is going to form networks to be responsible for the clot. You're not going to have to know these individual factors as to where they come into. These insoluble protein fibers form a meshwork, and it's going to capture all these cells, okay? Plug is formed, platelet contractile proteins pull the edges of the clot inward, stabilizing and squeezing out the serum. Swift removal of clotting factors by moving blood keeps clot from growing. You don't want it to be, you don't want to continue to grow, okay? You don't want it to trap up normal vessels. You don't want the clot to grow in the normal vessels. Get inhibition, get restriction of thrombin in the clot, antithrombin 2, and heparin from the basophils. These are just things that, just remember, the clot, once it's formed and does its job, is going to be deactivated, okay? Medical aim, these are anticoagulants. This is the old one, Coumadin, okay? This is what is in the purple top tube, EDTA, okay? And I don't know what CPD stands for, TPA. TPA is something they use for either in the heart or in the lungs when you get a pulmonary embolus or a cardiac embolus. Flavonoids, tea, red wine. Clotting disorders, you can get thrombocytopenia. Under 50,000, spontaneous bleeding. Bone marrow malignancy, treatment, transfused platelets. Hemophilia, you can be missing, you can have different kinds of hemophilia. The most famous one is factor VIII deficiency. There are, in the English royal family back in the day, and in the Russian Tsar family, one of the wives, hemophilia is a disease carried by the female. It's on the X chromosome. And if she gives that X chromosome to the male, but it has the hemophilia gene, because the male doesn't have a corresponding normal X chromosome for that. Male has, these are sex chromosomes, has a Y chromosome, and it's expressed in the male. So it's carried by the females, expressed in the males. And back in the day, the females didn't get hemophilia because if they had both chromosomes, they would die either during childbirth or during the start of menstruation, okay? Transfusion or injection of missing clotting factors. Thrombosis, intravascular clotting, usually in the vein. Atherosclerosis, or you get too slow blood flow, you can get an embolus. Blood groups and blood types. We are going to talk about this next time, okay? Because I want you to work through your blood typing and learn about your blood types before we talk about this, okay? And so we really don't have too many slides to go through. So next Thursday when we meet, we'll pick up with blood type and then go into the heart, because I want you to work through those exercises, at least through the one on Pearson, because that's due next Thursday, okay, on blood typing. So in summary, we did an extensive introduction to the course, okay. Most everything you're going to need is under starter course documents. We do it by the modular system. Announcements are very important, very, very important. Announcements are very important because they tell you where you're going and where you need to be, okay? This is an example of week 1. I showed you what week 4 looked like. This is all jammed up because we're literally just starting this. I'm essentially repeating the same stuff, okay? But starting with week 2, it won't be, okay? But this is your roadmap for the individual week, not your Canvas listing of assignments, but this, because Pearson is not hooked into Canvas. I don't hook it into Canvas. So Pearson's a separate thing. You have Pearson homework assignments, which we saw were 3 assignments: Introduction to A&P, the chapter test on 17, and blood typing, which are due next Thursday. You have 1 lab, which is chapter 17 lab module, which is found under Pearson assignments. It says lab, then say homework, and it's due in 2 weeks. Your labs are going to be due in 2 weeks. Your Pearson assignments are going to be due in 1 week. Email, use your school email. If you send it to Outlook, I'll get it right away. japhill@fscj.edu. I will get it right away. And then it will pop up on my Outlook and I can answer you and we can go on from there. If you send it through Canvas, I have a Canvas app, but I have to look at the Canvas app. So I look at the Canvas app a couple times a day, okay? Outlook, I look at it, it's right next to something important. I forget what it is, okay? Could be Instagram, I don't know, but it's right there, okay? I think it is Instagram. So it's, and I'm a big golfer, so I follow all the golfers on Instagram and a lot of golf stuff. And a lot of times when you send me a message back on Outlook, on Canvas, I don't see the message. I see the message, in fact, that it's there, and I see the first few lines of the message, but when I click on the message, I don't see it. And so then I've got to get up and go to the computer. And if I'm not so inclined, I'm not going to do it. Whereas if you send it to Outlook, which is what I request, I'll get it right away. I can answer it while I'm watching Young Sheldon tonight at 8 o'clock. Okay. I can answer your email while on my phone because just like you, I do most of my stuff on my phone. I ask you in the, it's the email policy, you're supposed to write BSC20860. You don't have to do that because I'm only teaching second semester. So that pertains when I'm teaching first or second semester, just write either Monday or Thursday. Okay. Excuse me. You're the Thursday class. In the subject matter line, write Thursday. I know exactly what class you're in. Okay. Because you can see the two classes are at different places. Okay. The two classes are in different places. They're going to be starting on chapter 20 next week. You're going to be starting on chapter 18. Now it won't be very long. It won't be very long. You'll be back on track with them because we all got to get to the same place by May the 5th, I believe. Okay. All your assignments are due by that last day, which is Tuesday at 6 o'clock in the evening. Okay. Because I have to have grades in that Friday. And most of you will be waiting on your grades so that you can see if you can get into the programs, or you can see you got to transfer to UNF or whatever's going on. Okay. And so everything's due. And the other thing is the late work policy. You will still get credit for it. But if you turn it in late, we're talking about the Pearson homework or the lab assignments, not the exams. And we won't talk anymore about the exams tonight because we don't have an exam for 3 weeks. But the Pearson homework assignments are the lab assignments. You will lose 2% a day, first day after you turn it in late, 2% a day, up until a maximum of 20%. Then it'll stop. So the maximum you can lose is 20%. So you can do it at the end of the semester if you want to, but you're only going to get, if you do the whole complete assignment, you normally would have got full credit. You normally would have got 20 points. You would lose 20%, so you'd only get a 16. And I illustrated the purpose to that for you. Number one, you need to know, chapter 17, big ticket items, blood typing, blood groups and blood typing. That's the big ticket item, chapter 17. Chapter 18, big ticket item is how does blood flow through the heart and what's the electrical system of the heart. Okay. There are other important things, but those are big ticket items. So if you don't do your homework, you're not going to get the benefit of that. And I can see that when we get to the exam, because I guarantee you there's going to be some short answer questions on it. I guarantee you with 100% certainty. And we will have a 30-minute review before the first exam. And I'll talk more about the exam as we go along. I'll talk a little bit about it. Anybody got any questions? We will probably never go to 8:30. Okay. My voice usually gives out about an hour and a half or 2 hours. Most of the other class, I've been getting done with the lecture by about an hour and a half. I left off the same place the last time with the other lecture. I believe we're pretty close to the same place so that you could do the blood typing thing. So it'd make much more sense to you after you had done the exercise on blood typing that when I talked to you about it. Because that is the big ticket item. Well, if you've got no questions.
Hi, I actually have one.
Okay, go right ahead.
Sorry to interrupt you.
You're not interrupting me. I tell you, just press your space bar and interrupt me. Okay, what's your question?
Okay, I just wanted to make sure that all of the labs are going to be through the actual online system, and we won't really be going over them on Thursdays.
We won't be going over them?
Like kind of. So like, like when you have like an in-person lab, especially a science one, they typically go through and they have like a pre-lab lecture. Yeah, they have prepared.
Right. Now, what we'll do is, because the first lab would be blood, and blood was looking at the blood slides and actually doing blood typing. That's why I put such a heavy emphasis on blood typing and why I put that worksheet up. So we'll actually be talking about blood typing next time. Okay.
Okay.
But looking at the peripheral smears, looking at that one picture that we saw that had all the cells that we wanted to see. Okay.
Mm-hmm. Okay.
So there really was nothing. So the answer is no, although I can't say that per fact because I no longer am able to do the PhysioX like I wanted to do, I had to put it through Pearson. And so I'm trying to move more lab into Pearson. I can't say
Right.
that we won't be doing something different than what I've done in the past, okay? Because I no longer
Okay.
have that access point of being able to get you to PhysioX just from following my instructions and you doing the lab, printing out a report and then uploading that. So it might be a little bit different. So I don't want to say, no, we won't be doing that, but certainly the things that would be emphasized in the lab, you know, one of the things that you're going to do for chapter 18 is you're going to watch the same 3 heart videos, the dissection of the sheep heart on Pearson that you would have watched before you came into the lab to do your heart dissection. Okay. So you
Okay.
watch the same 3 videos, but then the difference will be after you watch the videos, you will you'll fill in a diagram, okay, with the structures, and also you'll answer a few questions about the videos. So it's changed quite a bit. I do the best I can, and unfortunately, it's the physiology part. So physiology you can learn by actually doing the simulated blood typing thing. You actually can learn physiology by doing that, and it works out pretty good. Like one of the physiology things we'll be doing, looking at blood flow, if we change the diameter of the radius or if we change the length of the blood vessel. Okay, and you will see positively, without a doubt, unless you don't do the lab like I asked you to do, that the radius affects blood flow. Blood flow increases if you increase the diameter, and if you increase the length of the blood vessel, blood flow decreases. And so you learn those techniques by doing the lab. But primarily the anatomy and physiology 2 lab was, you know, you had your heart dissection, you had your kidney dissection, and then you had your blood typing, and then the rest of it was looking at models primarily. So that was primarily anatomy and physiology 2 lab. It wasn't nearly as hands-on as anatomy and physiology 1 lab was. It was doing a lot of modeling, a lot of models. So
Okay, that makes
I'm
sense.
trying to, when we have our meetings, that's one of the things that we talk about is how to best do the labs that the students can get the most advantage out of in life, that we're teaching the majority of these classes online. And we actually have Canvas shells, okay? We have Canvas shells where if somebody finds something really neat, they put it into the shell and they'll put it for a lab for this chapter. Okay, they'll put it for a lab for this chapter and they'll put it in the shell. And so that's one of the concerns with all the faculty, or the vast majority of the faculty that attend the monthly meetings, is what can we do to make the lab portion, which is where you lack out a lot. Okay, I mean, I enjoy face-to-face classes. I was a plastic surgeon. I'm a face-to-face person. Okay, I like to talk. Okay, and so I do.
Okay.
I like to talk. My wife, I've been married 45 years, and she doesn't like to talk. She's a nurse, was a nurse anesthetist. Okay, and she doesn't like to talk, and so I just talk to myself because she never says anything back to me. So I talked to the Great Dane. We got a Great Dane who was in here a while ago. Fortunately, she didn't disrupt everything. But my whole goal for everybody is to make the class where you can learn as much as possible and get your A or B. So if you want to go into nursing, you want to go into physical therapy, you want to go into whatever you want to go, you can go into it. I started as an OR. Actually, I started as an orderly. I worked as an orderly for a while, a couple of summers. I worked as an OR tech. I became a physician. I did 5 years of general surgery. I did 2 years of plastic surgery fellowship and 6 months of a hand fellowship as a plastic surgeon for over 20 years. In the process, I got my MBA, I got a law degree, and I got an LLM in international taxation and financial management. And I don't use any of them except for my medical practice and what I bring to you, because I will tell you, if you've had my class before, I say this is something you got to learn that you'll never use. Okay. And it is. It's something you'll learn that you never use unless you go into that specific specialty. Just like I mentioned that I didn't remember any renal physiology. I was a plastic surgeon. Okay. And I didn't remember any heart physiology. I was a plastic surgeon. So I had to relearn all that 5 years ago when I started teaching this. So my whole goal is to get you where you want to be. And I just wrote 2 recommendations for people, one for something in Gainesville for pre-med, some kind of summer thing, and another one was for scholarship. And I can tell you when I write my name and write MD, MBA, JD, LLM after it, it's got a lot of good initials, and I'm always willing to help you write a recommendation. If you ask me for a recommendation, 2 things you got to give me: your whole name, okay? 3 things, what you need the recommendation for, and you got to send me some information about you, either a CV, a resume, or a couple of paragraphs, essentially the introduction, okay? So I can say more than John Doe was in my class last semester. John Doe got an A. John Doe is in my class this semester. I mean, that's not a very good recommendation, okay? But if you tell me something about you, I can interweave it in the process and making it sound like, that there's some, there's somebody that's really all it takes. So if anybody needs a recommendation for anything, I'm happy to do it. You just got to send me that stuff and tell me what you need it, and tell me how quickly you need it. Don't tell me you need it tomorrow, unless you send a what is that, Vimo, PayPal, all that stuff with it. I'm just kidding about that. Anybody here?
I'm here and I'm listening.
Okay.
I appreciate your.
I'm kidding about the PayPal stuff. No, I'm happy to write recommendations. I'm happy to get you to your A or B if you just do the work. We'll get you there. Okay. Any other questions? If you don't understand anything, I'll have this video up by tomorrow. Oh, Florida Computer Nerd is my YouTube channel, but I will put this up. This video will be under module 17. It'll say recorded lecture spring 2021 Thursday. Okay, so you know it's your lecture. And the biggest part of the lecture part is obviously the announcements, the first part. But it should be up there by tomorrow. I just have to download it and let YouTube do whatever it does to it before I can upload it to the thing. You just click on it, and there'll be a little button to press, just like those other YouTube lectures, and you can watch it. Any other questions? All right, I'll see you next Thursday. Email me if you have any problems. Thank you.
Thank you, Dr. Phillips. Good night.
You're welcome very much.